Good morning, everyone. Welcome back to the Public Health Airlangga University Summer School 2022. My name is Erni Astuti. I'm going to be the moderator for this course today. Actually, Public Health Airlangga University Summer School is an international program hosted by Faculty of Public Health Universitas Airlangga that is aimed to engage global academic community. Okay, the topic of this course today, we have statistical genetic and population genomic. Okay, first of all, let's say thanks to Allah Subhanahu wa Ta'ala who give us guidance, happiness, healthy, and mercy so we can attend and participate in this special event. Before we start this course, I would like to welcome Dr. Muhammad Atoillah Iswandiari as Vice Dean of Faculty of Public Health Universitas Airlangga. Welcome, Dr. Ato.
Okay, thank you, Ms. Erni.
Yeah, welcome Professor Sim Sueling from Swaswi House School of Public Health, National University of Singapore as speaker today. Welcome, Professor Sim.
Introduction to the
Oh, hi. Good morning, afternoon. Okay. I'm not sure. Okay, maybe because it's a small group, we don't need to be too formal. I've just changed my title based on your title. So let me pull up my slides.
Okay. Prosim, before you started, maybe Pak Ato Ilah will have opening remark. Yeah. Okay. I'm continue. Okay. The next agenda is opening remark that will be delivered by Dr. Muhammad Ato Ilah. Dr. Ato Ilah, time is yours.
Okay, thank you, Ms. Erni, and good afternoon, Prof. Sims. Alhamdulillahirrahmanirrahim. On behalf of the Dean and all the Civitas Academica of Faculty of Public Health, Universitas Airlangga in Surabaya, I would like to send our warm greeting to Prof. Sim, who will deliver the very interesting and I think it will be our future in epidemiology and many disciplines. And this morning, we would like to thank Prof. Sims for delivering the guest lecture, entitled Introduction to Statistical Genetics and Population Genetics. I think it's a very interesting topics, and it is our pleasure and pride for having collaboration with South Hook School of Public Health from National University of Singapore, who is the ranked first of the university in Southeast Asia or in Asia. and for your information, Universitas Airlangga right now is at the rank 81 in Asia, according to QS World Class University. So again, welcome, and we are happy for having you here, and hopefully all the attendants will enjoy and will gain some fruitful discussion, a fruitful lecture, and yeah, maybe from this morning we will extend and enlarge our collaboration in the future. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Ato, for deliver the opening remark and also open discourse. Okay. For all participants, I would like to inform you that while the presentation is in progress, if you have any question, you can type the question in the chat box or later in the discussion session after Professor Sim delivered the presentation. You can raise your hand. Okay, here we have Professor Sim Swiling. Please welcome, Prof. You have 60 minutes to deliver your presentation. The time is yours.
Okay. Hi, everyone. 60. I prepared for 2 hours.
Oh, okay, it's okay.
Okay. But we can see how we go along. Okay. So what I have actually prepared for today includes some basic concepts of what gave rise to statistical genetics. And then we'll go through some of the changes and the evolution of how they have been applied to genomics in the last about 20 years. And then I will also then elaborate on what we call a polygenic risk score in more detail, which is currently the more hot topic in genomics now. So those are the 3 major sections that I've put through in terms of the talk today. Okay, so I first start, I think we know as we do genetic, we start with association studies if we were to do epidemiological studies. And simply, we just want to know if there's any exposure to risk factors, be it environmental or lifestyle, that could increase or decrease our risk for any disease outcome. We then go into genetic epidemiology, right, where we go into the genetic association studies. And now we have an additional exposure, which is our genetics profile, right? And our genome is a manual of 3 billion bases. And actually a lot between any 2 of us, we are largely identical. And the unique thing about the DNA sequence is that it doesn't change over time. Your exposure to your lifestyle environment actually change every minute, every day. Yeah, so the genome doesn't change, and that sort of made it sometimes a bit easier to study it than, you know, compared to things that keep changing over time, and you have to account for what causes the change. And the other major factor that allow us to do this kind of studies is technology advancement. Over the last 20 years, the technology advancement is the key factor that allow us to drive a lot of these factors. Without this technology, we won't be able to measure our DNA manual. So the history of human genetic studies, you know, there are a lot of different types of human genetic studies, right? From a long time ago, is what we know as the linkage studies, or sometimes, you know, the family studies, where you will study within a family, you will find the areas or regions in the genome that seems to co-segregate within a family for disease. So for example, we tend to, at that point in time, do trios, which is the father, mother, and the child. And we want to see if there's any, you know, any genetic that co-segregate with this family. And those are the early days for human genetic studies. Gradually, we started with, you know, candidate gene study. For example, we are interested in, say, ApoC3 gene and its impact on cardiovascular disease. Then we will look into this specific gene, right? So these are based on prior knowledge. It is only one gene, it is not the entire genome. And so these two are the early studies. Then maybe about 15 years ago, the first common variant genome-wide association studies came about. These are hypothesis-free, and it makes use of technology where we have highly accurate genotypes of about 100,000 to about 1.2 million from arrays from commercial companies. And then currently, what many people are also doing are what we call sequencing, which you will sequence the sequence of variants for each individual. This is more comprehensive. It can capture things that are more rare, because when you go on the array, it's a fixed set of contents that you put on the array still. So currently, what's more common is actually the array-based genome-wide studies and sequencing studies. Now, sequencing studies are still very expensive to carry out at 10,000 or 100,000 individual scale. So those studies are still at a smaller scale compared to genome-wide association studies. This was taken from a paper about 3 years ago. So progressively, human genetics, we started off looking at human genetics to understand the disease, to see whether we can get new targets for drugs. And then they are also used for Mendelian randomization because the DNA doesn't change over time, and it helps to understand causality. And in the last couple of years, it's also gone through what we call a stage where we have a term for it. It's called precision medicine, where the precision medicine has got a lot of more defined aims, where we want to understand progression. We want to understand if some diseases are different within the subtypes, and if we can make use of the genomics to inform whether an individual should receive a different type of treatment or specific treatment different from others. And these are what we generally coin under precision medicine. Maybe a success story from human genetics. This is the PCSK9 gene, where in 2006, a group found a genetic variant that seems to decrease the low density lipoprotein, which is the bad cholesterol. And with that, they actually took it to make a drug. And in, it still took about slightly over 10 years before the drug finally got marketed. But this was one of the success story for which a genetic finding actually led to a development of a new drug. So briefly, maybe I will catch up on some of the terminologies. For example, here we have what each, if you can imagine, each row represents an individual, and then you have the sequence of the genome. And here, the frequency is what we have at each base pair at each point in time. Alleles and what we call are different forms. For example, at the 5th column, the 2 alleles are A and G. And then we observe 4 copies of A in 10 chromosomes. And so the frequency of A, allele frequency of A is 40%. And that's what we call common. And usually we define common as when the frequency is more than 5%. The other example is where 1 copy of A is observed in 10 chromosomes, right? So the allele frequency is about 10%. Okay, so now I masked off the, I masked off the ACTG, right? And so each line is actually in chromosome, and the red circles denote the alleles that are different. So you assume that those with no red circles are the same. Now, a very important concept for genomics is what we call linkage disequilibrium. It essentially means the correlation between each pair of genetic variants. And it's a mosaic where some people look more similar to each other and some people not so. And the key factors that determine the similarities or the differences is based on mutation rate, the recombination mutation rate, which will increase the differences between people and decrease the correlation. The recombination rate will also decrease it because the chromosomes will get all mixed up as it recombines. Population size and natural selection. And natural selection also means that natural selection actually means that they will look more similar because for some characteristics, they tend to confer an advantage and then they will be inherited together. So in a bit more detail, we go back to the figure from before. Now, let's now label them as variant A and B. If we know variant A and we also completely know variant B, it means that the R square or a measure that we use to define linkage is disequilibrium is 1, which means that I only need to know variant A and I can know variant B. I don't need actually both variants to be present. In this example, if we know variant A, we do not necessarily know what happens at variant C, right? If you go back, if you look at variant in this example, anybody with a red circle in variant A will also have a red circle in variant B. Anybody with no circles will have no circles in variant B. But this is not consistent in the next example, right? There are these two people with circles and they have no circles at variant C, but it's not consistent throughout. And this is where we call low linkage disequilibrium, because knowing A does not give us much information about C. The other concept is about a genotype and a phenotype. In the previous examples, we have it by chromosomes and everybody has 2 chromosomes. So there are 5 individuals here with 10 chromosomes, 10 lines, horizontal lines. When we read across the 2 chromosomes for each individual at the same position, this is what we call the genotype of individual 5 at this position. When we read across the chromosome is what we have as a haplotype, which is a combination of the different alleles across the whole region, okay? So this graphically makes the distinction between what is a genotype and what is haplotype. Okay. Now, then we look at the differences between genotyping, sequencing, and a third concept, which is again a very important one in statistical genetics, and actually is one of the development that allows us to do a lot of these studies now, is what we call imputation. So genotyping means that we first design the array and the contents that goes onto the array. It can be 100,000, it can be 600,000, and then we measure it on our individuals. Then sequencing would mean more comprehensive. We get a lot more data. And then statistical imputation means that we have what we call a reference panel, which is a dense reference panel where we know a lot about the samples there and from different populations. And then we use them to infer the missing information based on linkage disequilibrium. So you can imagine that we will try to match our samples to the reference panels and match based on the haplotypes. If they are similar, we will then copy the missing genotypes to our samples. So the orange ones are what we call imputed or inferred from the reference panels. And it's a free way to get more data with very minimal cost. Okay, then once we measure the genotypes, basically what we do for association testing is not very different from epidemiological studies, right? We can have a case control design where the samples are ascertained based on a certain disease, and then you will have cases and controls, and those are usually done with a logistic regression. You can also have a what we call quantitative traits like say blood pressure or your cholesterol levels. And then we will identify genetic variants that have got differences in the trait means. And this usually is done with, say, a linear regression. And broadly, we define 5 times 10 minus 8 for level of significance. And this relates to a million independent tests. Okay. So going back into, what we have as the frequency of the variants, right? The allele frequency is on the x-axis. And on the y-axis, we're looking at the impact or the disease risk. Different types of study design allows us to do different hypotheses. For example, for those with rare and high impact alleles, these usually are for the monogenic disease. And then the low frequency and for intermediate penetrance are the ones that are actually quite difficult to detect, and it requires either sequencing or imputation from large numbers. The common variants are the ones that we have done relatively well through imputation. And what we have seen from a lot of the studies is that we mostly detect low impact common variants. And that's mostly what we are seeing now. Maybe I should use a pointer. Okay. Now I don't know how to move my slides. When I use a pointer. Okay. Now, we will start. So I said I will take you through some of the history and evolution. So we'll start with the early array-based GWAS. This was the first genome-wide association studies, or GWAS, that was published in 2005. And at that point in time, there were only 96 cases, and these are people with the eye condition, age-related macular degeneration, and they only have 50 controls. The array that they used had about 120,000 genetic variants. They reported a common intronic variant at CFH gene that crossed the significant threshold. And this is what we call a Manhattan plot. And it's called a Manhattan plot because it resembles the tall buildings that you see from the Manhattan skyline. The y-axis is the negative log 10 of the association p-values, which means the higher it is, the smaller the p-value. And on the x-axis is the chromosome positions of the genetic variants. Okay. Then about 2 years later, there was a set of 3 GWAS for type 2 diabetes in size. It's all done in separate groups. There's one in the Finns, there's one done in the US, and also includes some Swedish individuals. And these are from the UK. And what they coordinated to do was to look up their results in each other for confirmatory replications of their associations. Now that was in 2007. But after that, we progressed to use reference panels with imputation in order to perform what we call meta-analysis. So the first meta-analysis means that they were able to pull all the data together and not just simply look up the top 10 or top 20 hits in each other. They were able to use imputation to then enable the meta-analysis to combine the evidence across the studies. So this was in 2008, where the same group of people came together and they actually combined their studies instead of doing their results individually for the 3 studies that were shown earlier. They did they had different number of genotype variants. And so what they did was they imputed and gained quite a substantial number of inferred genotypes and then combined it together. All right. So this is currently what the norm is to do imputation and then to do meta-analysis, so that you bring together hundreds of thousands of samples or even up to millions of samples. But we also know that, populations are different, and largely there is a lot of studies from the European ancestry, and then gradually, I think in the last couple of years, there's a lot of push for non-European studies. But in the initial years, most of the publications were based on European-based studies. I think just to illustrate that further, so this is what we call a principal components map of your DNA. You can read this as the first principal component, which is on the x-axis, right? This first principal components separates out the African, right? Can imagine there seems to be a separation here. Separates out the Africans from the non-Africans. And the second, right, is also separating out the Europeans from the Asians and the Americans, okay? And as an example, the individuals from Singapore were mapped. For example, the Indians, who are actually migrants from South Asians, were mapped closely to the South Asians. The Chinese, who are also migrants from China, were mapped closely to the East Asians. And then the Malays are in between the East and South Asians. More similar to the Chinese than to the Indians. Okay, and so this is a map if you were to just consider, individuals within Singapore. Okay, now, as I said, right, this was another illustration that a lot of the, association findings, a lot of the genes that we reported, they were mostly done in European studies. As you can see, that overwhelmingly, there's still a lot more of the purple, which corresponds to the European ancestry. But this is quite an old-ish paper about actually about 6 years ago. It's largely improved with quite a number of biobanks around the world now. So what we learned throughout those years is that, different populations, they actually do share quite similar genetic risks to a lot of common diseases. But what we also notice is that because of the differences in the population history and sometimes environmental influences, which, for example, things like natural selection, the Europeans will be able to drink milk because they are lactose tolerant, and that has to do with a selection advantage and is reflected in their genome. And this can result in different risk profile across the populations, for example. So we also recognize that despite the similarities, there's always advantages to look into different populations. So just to illustrate an example, this was a whole exome sequence data where we collected individuals from different ancestry from around the world, and largely based on the major continental groups. So we had Hispanic, African Americans, Europeans, and then East and South Asians. What we notice is that a lot of the exome variants that we notice are rare and ancestry specific. So this is a stack bar plot of the variants, right, that we notice. And for the majority, or actually 3/4 of the variants were only seen in one ancestry, and about 1/4 or less than slightly than that in 2 to 4. And only 3% was seen across the ancestry. Now, when we look at them by, the type of genetic variants that they have, whether is it protein truncating, which means that it does change the amino acids at the end, Non-synonymous also change the protein acid, and synonymous means that the resulting protein is still the same despite the change in the bases. We start to see that the specificity comes from the coding variants. There's still, so then that still suggests that, different populations do carry different information. What we found from the paper, and this is what I mentioned earlier as a Manhattan plot. And increasingly, as we begin to find more associations, the previously known ones were colored in blue here, the region that we already know. This is a new one that we found at that point in time, where it is only seen in East Asians. Remember, there were 5 populations in that study. And so this was only seen in East Asians. And interestingly, this PHAUSS coding variant is only seen in East Asian and not anywhere else, right? And so it's actually really considered quite common in East Asians, but it's virtually zero in other parts of the world. And so because they had at least certain amount of carriers or frequency, we actually have power to detect these associations between PAX4 and type 2 diabetes. Okay. And we also saw actually that, before this paper was published in 2016, East Asian GWAS also found something nearby, a genetic variant nearby, but we weren't able to see these because we didn't have these in our data. It's not in our imputation panel. So the array-based plus imputation and the sequence-based studies, they still do complement each other because sometimes we do find things that's in one and not in the other. And then this PAX4 is not a completely unknown or it's not a gene that hasn't been implicated in diabetes. It actually has been implicated in some of the early onset diabetes and previously reported in a Thai paper, in the set of Thai, I think it was a case control. And it seems to have a role in the islet differentiation and function, which is relevant to type 2 diabetes. Then, so after that, in the last couple of years, I think in the last 5 years, we now have this new term called trans ancestry, where we now combine different ancestry together. The left is one that is the published for lipids, for cholesterol lipids. And on the right side is one that we recently did for type 2 diabetes, where we combine, again, we mostly look at the 5 continental groups and we combine the data across the 5 continental groups or other groups who contributed the data. So we go back to sort of this map, and I wanted to go a bit more about this clinical heterogeneity and a bit more about this polygenic risk score. And this polygenic risk score sort of gained traction recently because we start to find a lot of common and a lot of low impact genetic variants associated with an outcome. So, at this point, I think we have more than 400 regions in the genome associated with type 2 diabetes. And can we then use a summary score to summarize the overall impact of these 400 variants and calculate a single value for the risk of that individual for type 2 diabetes? And that's the broad concept of a polygenic risk score. Okay, and so this is what I said before, right? The per variant effect size is small. And it doesn't explain too much of the outcome variability. And it gets even smaller as sample size increases. We recently had a publication for height that went to more than 1 million individuals. And so the question that we're asking is, based on your whole genomic profile and not just based on the 1, 2, 3, 4 genetic variants, what is the cumulative effect of your genomic profile? Okay. So conceptually, we started off with what we call a genetic risk score, where it was done actually even before now, where you collapse the contribution from multiple independent genetic variants and associated with a specific outcome. It is commonly used for, say, risk prediction. If you have a higher score, do you have a higher risk? There are various methods of computing this, which I will show later. And most of the time we would use the rescaled weighted genetic risk score. So let's go through some simple toy examples for computing and understanding what a genetic risk score is. We have here in this example, summary statistics that we get from large data analysis. We have 3 variants here, and then we have the effect size. So we know which ones confer, say, increased value, for example. And then we have test data. This is our test data with these 3 same genotypes, right? So what we end up doing is we will have, for the first individual, right, we will calculate. So let's say this method is just summing the number of risk allele that the individual have. So the first person has an AG, right? And that means that the risk allele is A. So this is 1 copy of A, right? For the second RS456, the risk allele is T, right? And this person has got 2 copies of T, so it's type +2. And then for the last one, the risk allele is T, and this person has got 2 risk allele T for RS789. So for the first individual, he or she carries 7 copies of the risk allele out of a total of, what is this? Sorry, this should be 5 out of 6. I must have had another variant that I took out. So this should be 5 out of 6. Okay. The second one would weight it, right? So instead of just summing up how many do you carry, you weight it by the regression risk effect sizes. So then you have 1 copy of A, and the effect is 0.02, you times 0.02. Likewise, 2 copies of T times the risk effect 0.04, and 2 copies of T at the RS789 times 0.06, and we sum it up, right, to generate a single value for the first individual. Okay. And finally, one that is sort of rescaled means that we take what was done earlier with the weighted score, but we rescale it based on the risk effect size, which is we divided by the total sum of the risk effect size, and then we times 3 because there's 3 genetic variants. So these are the common ones where we would consider to collapse the cumulative genomic profile for the individual. We see that there is a limited predictive ability for genetic risk score. So this is an example where there are 32 BMI associated loci to predict obesity in almost 8,000 individuals. And you can see that the specificity and sensitivity does not increase too much, right? Compared to having just the, I think the colors of the dots corresponds to the age, right? The age that's presented here, okay? Then, so this one only considered 32, right? Can we do better, right? Now that we know so much more. So this one was another example where, even though they only use 29, but this is what we can use to illustrate the heterogeneity, say, between diabetes individuals. So this one used the type 1 diabetes. Now, type 1 diabetes and type 2 diabetes are actually quite different. So type 1 is more autoimmune based, and a lot of the genetic associations are found at the HLA region on chromosome 6 that are relevant to immune. And so when you apply that 29 genetic variants associated with type 1 diabetes across all diabetic individuals, you will see that the scores can separate those with type 1 that were diagnosed as type 1 and type 2 in a bimodal fashion, as you can see from the top plot. Okay, so there are some individuals possibly, right, where they manifest a lot later compared to what someone with classical type 1 diabetes will manifest, usually before age 20. So with a 40-year-old type 2 diabetes who have rapid progression, the rapid progression sometimes may make the symptoms of a type 1 diabetes possibly be a late onset type 1 diabetes that was missed, right? So this was one example where the outcome was very specific, in the sense that the genetic variants were able to separate them, different clinical manifestation quite easily, even though they only use 29 variants. So the utility of this kind of aggregated score does also depend on the disease architecture. Okay, so we described what a genetic risk score is. Then the new term is what is called polygenic risk score, or polygenic score, where they now try to get the cumulative information from the entire genome and not just from the specific independent variants. And that's what differentiates this from the genetic risk scores and the polygenic risk scores. Okay. The polygenic risk scores, of course, is a lot more complex. And so this is one of the quite a neat review paper that illustrates some of the processes to generate this kind of polygenic risk scores, which essentially you need different data sets, and they need to be processed. And then the calculation of the polygenic risk scores, there are different ways to do it, and then you apply it in a separate data set. This to illustrate. So for example, we'll start off with these summary statistics, right? Again, not very different from the genetic risk scores. And the target data is the data set that you want to calculate the polygenic risk scores on. So this is not very different from the previous example where I have 3 SNPs, RS1234, RS45123, RS456, and RS789. And these are the 3 individuals. Okay. Because it's now genome-wide, so you're looking at a lot more variants, and there needs to be a lot more stringent quality control to make sure that you don't include too much noise into the data set. And then you will retain the data that overlaps between this base data and the target data. To calculate polygenic risk scores, there are different methods that have been used. For example, one is where you will have to reduce the LD, which means that instead of 1 million genetic variants, you will have to reduce the LD such that the correlation between neighboring variants should not be more than, say, 0.6, right? So then that may reduce the data set to be about 600,000 instead of 1 million. And then there are also methods where you shrink the coefficients, right? For those genetic variants that are highly correlated, I only need one, I don't need the others. So then you will shrink the coefficients of the others to 0. And another easy to implement one is where you will just do the cutoff at the Manhattan plot and say, okay, I want to cut off at 10 minus 5, and I only take the genetic variants that show association less than 10 minus 5, and that reduces the number of variants that goes into it. Okay, so these are the common methods of generating the polygenic risk scores. And then once you generate these scores on your own target data set, you perform the association testing with the outcome of interest. Okay. There are a lot of work going into polygenic risk scores, the methods development, right? Including how do you account for the fact that different populations may have different scores. And there are also different ways to look at the scores. Now, this is one example where there are a lot of, quite a lot of association for type 2 diabetes, but we know that some of them work through different types of pathway. So there are some that are because of the lack of productivity of the pancreatic beta cells. There are some that are because of obesity and the fat stored. So this is an example of it being applied where the clustering of it happens within 5 major categories of what they consider as major contributor to type 2 diabetes that includes obesity and the liver function. Okay, and they were also able to use the scores, right, to represent each of these pathophysiology subset. Another example is that you can use a polygenic risk score to do what we call a phenotype-wide association studies, or PHAUSS, and this is one of the example from a cancer example. So it was meant to understand what is shared and unique between different disease subtype. And so what you see here is 3 examples of 3 common skin cancer where you can perform the polygenic risk score generated from one of primary skin cancer with the other data that is collected here. So for example, you have polygenic risk scores from squamous cell cancer, and then you apply it through the other data or the other outcomes in the data set to see whether the polygenic risk scores also is associated with any other outcomes besides squamous cell cancer. This allows us to see if there is any similarities or shared genetic architecture between them. Okay. As I said, there's a lot of work going on to polygenic risk score. It's not quite as robust. And we also ask the question, is it ancestry specific? Do I need to generate, summary statistics for, say, East Asians just to generate polygenic risk scores on East Asians? And the differences between the populations and the ancestry, are these true differences for outcomes, or are they just underlying genetic differences between the populations? Okay, and at that point, what we saw was that the performance of polygenic risk score that is derived from one ancestry tend to have poorer value when applied to other ancestry, and this is especially true when you apply on the African population. So this is another example, right? We're looking at height here, and the polygenic risk scores can vary very differently across the different threshold. And then you have, again, the 5 major ancestry, and these were applied across different data sets. And we can see the curves, the curves actually do not overlap. So for the same traits, the polygenic risk scores can vary quite differently across the different thresholds. Okay. And of course, the other problem, as we have seen before, is that a lot of the studies, the ancestry of the GWAS participants are still predominantly the European ones. And so, it's a little bit disproportionate in terms of the information derived from the non-European participants. And so what the group noticed, so they actually had 3 biobanks, from European, Japan, East Asians, and Africans, and they tried to cross-relate the summary stats across for 5 disease subtypes, 5 phenotypes. And what they summarized was that when you match the summary stats to the data that you have, the predictive and accuracy is always higher, and it's almost always poorer for Africans. Okay, so this is what they found at that in the publication about 3 years ago. But recently, the last major paper on meta-analysis of lipid traits found that if we were to use multi-ancestry polygenic risk scores, when we combine the information from the different populations to begin with, when you apply it across different populations, they tend to perform similarly, although they will still perform slightly poorer in Africans, but for the other populations, they seem to do pretty well. So, for example, this is the African panel, right? And they had polygenic risk scores from everyone, all polygenic risk scores from European and admixture Africans. You'll see that the rate from the admixture populations will always have the highest adjusted R-square, followed by all, which combine information across all ancestry. And the poorest one is the European only derived summary statistics. So if we look at the Europeans, right, the Europeans and all perform well, whereas the African one do not perform as well. So either you have it as ancestry matched, or you should generate the polygenic risk scores from everybody so that you can have as high an adjusted R-square as possible. So then, of course, the question is, we can estimate polygenic risk scores. How can we use that, say, in the clinic, right? Is it helpful when we compare it to, say, things that are monogenically inherited? How about when we compare it to, say, a common trait like BMI or lipids? And is it more informative than when we ask somebody, do you have family history of diabetes? So some studies have shown that incorporating polygenic risk scores can have better prediction of early onset cases. Now, these are, coronary heart disease, type 2 diabetes, atrial fibrillation, and then there's 2 cancer below, the breast cancer and prostate cancer. And this shows that people with higher polygenic risk scores, they do have increased risk over time as age increased. And, if we, and you can calculate polygenic risk scores at any given age, right? Because it doesn't change with age. We are able to generate polygenic risk scores for each of these individuals and possibly be able to predict early onset cases, right? So if somebody falls in a higher polygenic risk scores for breast cancer, would that be a reasonable case for somebody to recommend more frequent mammogram screening compared to somebody with a lower polygenic risk score. They also show that the utility of polygenic risk scores, they can differ by the outcome, right? For example, it can differ between the cancers and the non-cancers, right? So there may be a high inheritance for atrial fibrillation or for early onset and prostate cancer. But that's not quite the case, when you look at late onset, where for breast cancer, the high polygenic risk scores actually made up a higher proportion of the individuals. So again, the utility of these polygenic risk scores do depend on what outcome you're looking at and what the original genetic architecture of it is. Okay. And another study also showed that if you include polygenic risk scores, you can improve the classification of, say, prediction. This is 3 examples of heart conditions, and they had prediction matrices, the discrimination matrices. They had conventional risk factors. They included the polygenic risk scores, and then they also included one more biomarker, C-reactive protein. And you see that with respect to the conventional risk factors, the inclusion of the polygenic risk scores does have some improved classification, say for coronary heart disease and in general cardiovascular disease. Okay. So that's how some of the polygenic risk score is being used, or how many people are approaching it for now. It's not exhaustive. That said, I illustrated polygenic risk score a bit more because it follows pretty naturally from genome-wide association studies, what we would do from the associated results. But in essence, actually, it's a much more complex kind of a research world than what I have here. For example, we are only here, the GWAS signals is only here. And then you have the associated regions, right? And then you have these candidates where you have to figure out, right? Are these candidates going to be functional? Would you bring it to, the labs, right? And there's a lot of work that a whole bunch of people from different domains are working before it gets to a functional candidate. So I guess you can imagine, the PCSK9 going through the process up to here, whether it's functional candidates for it to be taken to, the biochemistry process of being made into a drug. And then, on the other hand, for the signals, right, for that genetic variant that we find, is it more likely to be the causal one? And more of the time, what happens is that it's in a region where there's no information, right? It's not clear whether it's coding. Coding variants are a lot easier to go. The path is a bit shorter if it's a coding variant to go to a functional candidate. But if not, right, if not coding, there needs to be a lot more work to determine whether it goes to a functional candidate kind of process. And then on the right hand side, there's a lot of biological work that goes into this, the experimental studies. And then a lot of work also goes into looking at whether some pathways or some genes are more similar to each other, or some genes are co-expressed together, and would that impact certain outcomes? And so there's a whole lot of spectrum that goes on through this. And for today, I've only shown you some of these from the GWAS signals. And definitely, there's also what we go beyond genomics, where we look at the proteins, we look at the metabolites, we look at, say, DNA methylation, to complement and to build a whole picture of what we think the genetic variants will lead to. Okay, so those are, you may hear them commonly as the multi-omics studies. Okay, actually it took me an hour. Okay, so to summarize what I have shown you today is that after so many years, current human genetic studies, they do include quite a lot of samples and substantially large. And especially now that there are a lot of biobanks in the world with high resolution. We also learned a very important lesson that population diversity is very important. It not only enhances the new discovery, but it also highlights, what could result in the differences, say, in the different prevalence of disease across populations. And then we also went through a bit about polygenic risk scores and what it can do on top of conventional biomarkers. Although it's still a lot of work in progress before it can actually be used in the clinic. And so actually, this is my last slide. Should I stop share and take questions? Okay. All right.
Yeah. Thank you very much, Professor Sim Swiling, a very wonderful presentation. Okay, for participant, if you have any question, please raise your hand.
If you like, I can send you my slides after this.
Yeah.
Okay.
Okay. Yes, please, Pak Rohim.
Okay, thank you very much, Bu Erni, and thank you very much for Team Professor Sim Shuling also. And I'm very interested with your presentation about the risk factor of DNA. Yeah, risk factor of DNA. And then maybe you answer me about some factor that affect to methyl DNA. Now I interested with research about methyl DNA.
Methylation? Is that?
Yeah, methylation. DNA methylation.
DNA methylation. I see.
Methylation. That's a very environmental factor to affect to methylation DNA. Maybe you give me information. Maybe you have research about it.
I see. Okay. DNA methylation, I think belongs to one of those where, as you rightly pointed out, is influenced by the genetics. It's also influenced by the environment, right? And so when it happens like this, usually, I run away from it a bit more. I've done 1 or 2 studies about this, and if you have seen examples, there are examples about smoking affecting the DNA methylation. And what we found is that, say, you can also generate DNA methylation risk scores with respect to, say, any outcomes, like what you would do for genetic risk scores, because they also act in the pathway to the outcome. But sometimes what we found, say, DNA methylation is actually an intermediate. So we were looking whether, does DNA methylation change your BMI or your BMI change your DNA methylation? And what we found was that actually the DNA methylation was changed by the change in BMI. So it's BMI first, DNA methylation, and then possibly downwards towards, say, diabetes, for example. So if you imagine genomics at the starting point and then your outcome at the ending point, DNA methylation actually is in the middle or somewhere, and it can be influenced by a lot of intermediary exposure. And that's the difficult part in some of these, in figuring out who changed who. So for DNA methylation, of course, you can also do methylation quantitative trait analysis, where you take every CPG sites and then you do association with your genome, right? Which part of the genome changed the CPG sites? And that computationally is very computationally heavy. But I've seen people do that and correlate the methylation change and the genetics with expression change. You can imagine everybody overlaps and see where the peak is. So those are, I think, some of the examples that I know for DNA methylation, at least the very few examples that I'm familiar with. I don't do a lot, but those are a few examples that I know that I'm familiar with.
Okay, thank you. And maybe island DNA always connected with DNA methylation.
Sorry, which
Yeah.
one?
Yeah, allele DNA. Yeah, allele. Yeah,
Allele
DNA.
DNA.
Always connected with DNA methylation.
It's so the DNA methylation can be changed by your genetic profile. And so that's how we assess with how it changes. Because different genomic profile can also influence the CPG site changes. So you can imagine DNA methylation is affected by both the DNA and what your environmental is, right? So those are the two components, right? So we can find the relationship between the DNA and the methylation. The harder part is to figure out how much of the environmental component goes into it.
Okay, thank you very much.
Thank you for your question. Hope it helps.
Yeah, but I also continue my question about the maybe all of exposure in environmental, yeah, chemical exposure in environmental that affect to DNA, also can become DNA methylation. Yeah. 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 All of chemical carcinogen. All of chemical carcinogen in environmental working, for example, can affect or become to DNA methylation. Yes. For example, just you teach us about a smoker, smoking, for example.
Smoking, yes. Smoking was the one that I read before, so I thought that that one was quite a classical example. But yes, I think, I've heard Bailey mention, carcinogens actually manifest a lot more compared to, say, the common disease like the heart disease, the diabetes and stuff. I think that's why that's why the outcome is very important when you study this, the impact and where it affects. For if we look at the lipids profile, it may more likely be for cardiovascular outcomes per se. And for DNA methylation, it may be more relevant or a bit more relevant to a lot of different studies than another set of studies. But at this point is at least what we know, right? We don't know until we do the experiments.
Okay, thank you.
Okay, the next question is from Bu Nur Mufida. Please.
Thank you so much for the opportunity. Let me ask about GRS and PRS, genetic risk score and polygenic risk score. Sometimes I find studies that use GRS and sometimes with PRS, and it's I think that it's different amount of genetic linkage that counts in the GRS, and it's different amount too in the PRS, and sometimes it includes 32 genes and sometimes include 29 genes like that. But I don't understand what is the causing of these differences in this amount of gene that include in this studies, and is that PRS is almost same with GRS like that. I think that I need more explanation about it, because which one is more sensitive for us to assessing this genetic modification. Thank you so much.
Okay, that's a very good question, because everybody gets confused and actually people use it interchangeably. The early days is genetic risk scores, and they would take it from independent. So which means the LD is minimum, right? So you will have maybe less than 100 in general, or at this point in time, say for diabetes, we have about 400 independent loci. Then you will have about 400 genetic variants that you include in your GRS. The formal definition of a polygenic risk score means that you include the entire genome as much as possible. So which means that instead of 400, you will actually include about 100,000, because you will assume that it's going to be like the case of, as your sample size increases, your power to detect small effects increases, you will start detecting a lot more of these small effects. And what happens is that the polygenic risk scores usually in formal definition should include at least more than 10,000 genetic variants in the score that you derive from. And if you look at these kind of methods to generate polygenic risk scores, for example, say if I do a p-value thresholding at p-value of say 5 times 10 minus 5, I may get about 30,000 variants that's less than that threshold, and I will end up with a polygenic risk scores derived from 30,000 variants. So I think sometimes it's used very loosely in terms of polygenic risk scores, and we really define polygenic risk scores as much as possible to include the entire genome beyond what is only associated. So genetic risk scores only take those that are really associated, must be associated, whereas polygenic risk scores accumulate over the entire genome. And usually at least 10,000 by rule, at least what I've started to see. So I think it's not so much you, but the few, sometimes I've seen a lot of people get confused when the term comes in. And when people use it loosely, right, when actually it's a genetic risk score, but they call it polygenic risk score, or vice versa, it confuses people. So polygenic risk scores, you can take it that it's not just the independent associated variants, whereas the genetic risk score is. And the genetic risk score is a very old traditional term that has existed a long time ago, and the polygenic risk scores is actually a lot more recent. It came by because now we have so many associations. It also came by when people start to know and understand a lot more about the genome, and they come up with methods to incorporate the entire genome. Right? In the early days, we were all afraid of overdoing it because we needed to account for LD. But now they know how to account for LD, and so we are now actually trying to be a bit more progressive to include the entire genome. Hope that helps.
Thank you so much.
Thank you.
Okay, for the participant, if any question, please raise your hand. Any question again, please?
Should take the chance to change this before I send this to you.
Yes, please. Any question? for the other participant? Oh, okay. Pak Sikit, please.
Okay, can you hear me, Bu Erni?
Yeah, I
Yeah.
can hear you.
Okay, first of all, I would like to thank to Prof. Sims willing to come to Universitas Erlangga and join the lectures this morning and giving many ideas for us, particularly for the researchers and lecturers that they have interesting for building research in the genomic data. So actually, I would like to give some just comment or maybe just give like a feedback for the Prof. Sim's presentation today. For the first time, so the genetic risk score that you have already worked a lot with the genomic data. I have question for your experiences. So what the performance of the genetic risk score or polygenic risk score compared to the conventional prediction models, like we just only have taking care with the other predictor like conventional biomarker and also some social determinant of health as the predictor. And when it is combined with the genetic risk score, I saw in your presentation that it is improve the prediction models accuracy, but it is not much higher, right? So if we are consider about the statistical knowledge, let's say this is not much any statistically significant. So this is the question that I would like to ask. So how to enhance the genetic risk score or polygenic risk score when we want to take prediction models, particularly for some disease like atrial fibrillation, type 2 diabetes, or any other other disease that you mentioned it before. Because actually we have a lot of prediction models in the literature. When we conducted systematic review, maybe we will have some models that already developed it. So my questions, is there any possibilities as well to do the validation for the genetic risk score that you have been developed, or you already validated the model as well? Thank you, Prof. Sim.
Yeah, I think that's a multi-million question that everybody is trying to do. So I think, firstly, comparatively from the traditional biomarkers, which you can get very easily, right? For example, I asked a patient, do you have family history of diabetes? Versus doing a whole array that costs you, I don't know, $200, 100
Yeah,
plus
this is
US
costly
dollar.
as well.
And then you have to still computationally process the data and then generate the polygenic risk scores and then to incorporate into the medical records to tell the doctor that this is the person's score and maybe this is the higher risk and build the model. At this point in time, personally, I think it's not worth the trouble. I think what you tend to see is that there will be some disease that will benefit more than others, right? For example, if they found for I think there are some for like I haven't seen. I mostly work in diabetes, cardiovascular. I've also seen some folks who are doing a lot for polygenic risk scores for breast cancer. Because if you have the score, you can do more preventive work with more mammographic screening. Instead of 1 year, maybe these women need to scan 6 months. And so if you ask me, I think it still has a very long road to go. Primarily, we are not at the stage where, it's easy for us to prick blood and then generate the polygenic risk scores. And that actually needs a lot of setup, right? And I can, I think, like what we did with imputation, right? Imputation in the olden days, you have to download the reference file and then use a lot of computers to impute yourself. 10 years down, somebody has a cloud, you just upload and then download, right? And everything is nicely done for you. There will be advances, I think, that will get us there, but we're not quite there yet. I mean, that's my sense. I don't know if you feel the same way.
Yes.
There's a lot of models, as 10,000 models. Even without the genetics, there's a lot of different models, right?
Right.
But what we've sometimes seen is that for certain things it does improve, for certain things it doesn't, and I guess the decision is, where do we decide that this is good enough for us to go to the next step? That's my sense. I'll be truthful that say I don't think it's there yet.
Okay, so about the genetic risk score that you have been developed, have you validated with another subject, like you did to another sample size that come from maybe similarly from ASEAN's population, but not having done in Singapore only, Prof. Sim?
I think most of them are transferable, and so we end up I mean, we have not done as much, to be honest, on the polygenic risk scores, because we also, the problem is we use a lot of our data to generate the summary statistics, right? So I need to wait for new data to generate before I can apply it to other people. I also need to wait for the outcomes to come. We haven't applied too much, but we do see the same things as other people. If you do East Asian and East Asian, it do very well. And then if you do everybody combined on East Asian, it also does equally well. So actually what we learn is that the transferability is actually very consistent across the populations.
Okay, so this is very good idea. If we have some models that can be applied in any race or any population, something like this. Okay.
Yeah, so I think currently the recommendation is good to have as much information as you can have for your own population.
Okay, very nice information then.
Thank you.
Okay, for participant, any question again, please raise your hand. Okay, I think there is no question again. We have come to end of the
wait. I have one question left.
Okay, Pak Sigit.
So the last question. So when you developed the genetic risk score, have you used like a machine learning algorithm to develop the models or not?
Not really. We mostly use a lot of LD pruning methods.
Okay.
I'm not as familiar with machine learning methods, but I'm sure there are a lot out there. Essentially, if you go and dig, right, there's probably 100 different ways of computing polygenic risk scores. The machine learning, I mean, it depends what you define as machine learning, right? Does ridge regression define as machine learning, for example?
Yes.
They mostly work on the principles of reducing the dimension and maximizing the information out of the minimum number of variants that you can get.
Okay. So this is very interesting one that particularly for me, because I'm in the division of biostatistics and population, and we are still developed about machine learning. So it is wonderful when maybe here we have a Dr. Diah Indriani as well, that in the future maybe we can collaborate and elaborate more about the knowledge that you have been sent to us about the genetic risk score, and then we can like develop the models or evaluate the model that have you been derived.
Yeah.
Okay, thank you. I think it's enough for me.
Thank you.
Bu Ernie.
Okay, thank you, Pak Sikit. For a student, any question again?
There seems to be one more question from.
Oh,
Yeah.
Ibu Mufida. Okay, please, Bu.
I'm sorry, I missed some questions. Only one, Prof.
Okay.
It's about can GRI's genetic risk for results describe LTL, long telomere length?
Sorry, again?
If the genetic risk score can describe long telomere length, LTL.
Yeah.
Is that it can describe about LTL? As I know, LTL can
Yes.
with many
The principle of polygenic risk scores applies to any outcome. So once you have enough associated variance with telomere length, you can generate polygenic risk scores. So it's a concept that's applicable to any outcomes.
LTL shortener or others.
Sorry? We lost you for
LTL.
Huh?
Hello? Include LTL.
What is
GRS
LTL?
can describe LTL. Long telomere
Yes.
Yeah.
So telomere.
I found telomere.
Telomere length is like BMI, height. It's just anything, right? So it can be applied so long as you have the genetic associated variants. And I know that there's a lot being published now because everybody is interested with telomere length with aging.
Yeah. Okay. Thank you, Prof.
Thank you.
Okay, thank you very much, Prof. Sim Swiling. It's a wonderful discussion. Okay, we have come to end of today's event. Hopefully this joint lecture can increase insight, relation, and other collaboration in the future. Once again, we give all our appreciation to our great speaker, Professor Sim Swiling. Thanks all students too. I hope you can get the experience in this course. And please don't forget to fill out the complete this attendance form. The committee just put in the chat box. Thank you very much. See you in the next activity.
Thank you.
Bye-bye. Prof. Sim, can you send the material? Oh, okay. Prof. Sim. Okay, see you in the next agenda. Bye-bye.